Hello, everybody. Thank you for joining us today. We have another one of our live virtual STEM classrooms. This one is for grades 7 through 12. It was put together by two incredible people joining us here today. Our education ambassador, Jen Cotton, and from WCS, World Wildlife Conservation Society, Dr. Harley Newton. She is a collaborating scientist with OCEARCH, and she's the veterinarian who's on the ship with us, on the platform with the sharks, while we are doing all of the work that we do. So she's a very important person to have, and she also has some very exciting work that she's doing that she's going to share with you today. Jen and Harley, how are you guys today?
Doing well. How are you doing?
Good.
Everything's good here in New York.
Awesome. Well, I appreciate you guys being here. I think you two put this together. So, Jen, I think I'll turn it over to you and Harley. How does that sound?
Sounds perfect. So today we have a very exciting presentation. Harley is a veterinarian. So one of the biggest questions we get asked pretty often is, what kind of careers can I go into in order to work with a organization such as OCEARCH? And this is one of the ones that I think people don't really think about, So we're going to look into what it means to be a veterinarian working on the list with these sharks. So Harley is going to give you guys a really detailed introduction into her career, and it's really awesome, all the things she does. So we'll go ahead and get started on the next slide just to give you an idea of some of the things that she's going to be going over with you today. So we're going to be talking about what health actually means. Looking at different tools that a veterinarian would use, and also talk about the different studies that they go over. So on the next slide, we're going to look at what health actually means. So Harley, if you would like to, you can go ahead and explain a little bit what it means for health in your field.
Sure. Yeah, the overarching goal today was really just to introduce you to health and how it can be used as a field and conservation tool, because I think most of the time when people think of veterinarians, they think of their dogs and cats, and maybe if you have horses, domestic animals, but they really don't see it as kind of a wildlife tool. So I just wanted to show you how we can apply it in the field. So health, most people think of it as being, the absence of disease or just not being sick, but the definition that we have for veterinarians is a state of physical and psychological well-being and productivity. So basically being physically well, being mentally well, being able to reproduce. Some additional things that we consider is also the ability to adapt. So being able to adapt to situations. So if you get sick, being able to get better. The things that we use, however, are largely for domestic species. It's things like, are animals eating enough? Are they going to the bathroom normally? Do they have a normal body weight? Are they behaving normally? Are they reproducing? These are all things that are next to impossible to monitor in sharks. They live in the ocean. They dive to depths of 800 meters. They're way offshore. These are all things that we can't see. So how do you answer questions about health in a species that you can't look at the things that we consider important? So we can go to the next slide. And it's important to differentiate health status not just by population, which is what we're doing a study on, but also individual animals as well. So in marine species, we really can't rely on sort of those outward physical and behavioral changes since they're hard to observe. And the health of an individual animal actually does influence its response to handling. So how healthy an animal is might impact how it deals with being handled by us on the lift. So it's really important to look at each individual animal. Population health is just absolutely critical for really understanding the overall effects of environmental and human stressors and all the environmental change that's going on. Underlying disease issues and other factors, they can impact populations on the whole. It might be that there's a species that is really depleted and they're not responding appropriately to all the measures you're putting in place. Are they a healthy population? Do they have diseases that we don't know about? These are all critical things that we need to answer.
So something that is key for science is using different tools. And the very first strategy is, making observations. So what types of observations are you guys looking at when you're doing your research?
Yeah, so the one of the most easiest tools to use in science is just look with your eyes. So the kind of things that we look for in animals is, how are they behaving, and are they using their habitat appropriately? If they're supposed to hide, are they hiding? Are they interacting appropriately with other animals in their environment? Are they active in the way they should be? If it's a species of shark that's supposed to swim fast, are they swimming faster? Are they at the surface and kind of at a strange angle? How are they swimming? All shark species have different swim patterns. Are they swimming correctly for that species? Are they swimming in circles? Other things are things like orientation. So like, are they swimming, across the water column normally, or are they tilted in either direction? Are they sitting on the bottom and they shouldn't be sitting on the bottom? And finally, breathing rate. That's one of the things that we monitor in, mammals and humans. You can, in some species, see how fast they're breathing and count that rate. So they're all observational tools we can use. The picture I have on the right for you is actually a photo that was sent to me by a collaborator, Toby Curtis, of what wound up being a mako shark. Mako sharks are supposed to be way offshore. This was a sick animal that was right near the coast, and that ultimately went on to die. So, this was an important observation, that it was in the exactly wrong habitat. So next slide.
And these are the cool ones. So this is what you're doing on the lift, right? So I'm going to actually do a physical exam on the fish. So go ahead and tell them some of those really exciting things that you guys do out there.
Yeah, absolutely. This is like completely a team event. There's no one part of this that I do all by myself, and everyone kind of has a job when we're on the lift. And we're doing that both to see if there's any health abnormalities in the animal, but also to monitor how they're doing when we're handling them. So we do get basic measurements. That's important because we don't know a lot about white sharks. We don't, we need to know how fast they grow, and we use those measurements to inform that type of information. Skin color is one of the few things that we can look like. If animals are really pale or they're turning really, really red on their belly, those can be signs that maybe they're not doing well. I will say it's pretty normal for a shark when they're being handled to kind of flush like a little bit pink. But if they get really dark red, that's one of the things that we're looking for to kind of signal that maybe we need to stop. Muscle tone. Are their muscles loose and relaxed, or are they really tight? Reflexes that we look for. So the two big reflexes in sharks that actually Brett monitors, because he's up at the head and he's monitoring the hose, is he checks their eyes for a reflex. So in species that have kind of a third eyelid, they'll raise it up. And then gill reflexes. If you tap the gills on a shark, they'll clench them. So those are some things that we can look for while we're on the list to make sure they're doing well. The other thing we can look at is just the vent of the animal at the back end, and just make sure that it has the right tone. So those are the things we look for in a physical on a shark.
All right, so again, you can just go ahead with this one with the body structure. So what kinds of things maybe would you use for looking at mass and the girth and all of that?
So yeah, the big problem with white sharks is they're really big. So it's really hard to weigh them. And that's usually how you determine whether an animal's healthy. Is it at the right weight? So the types of things that we do to kind of get a sense of how they are, how their nutritional condition is, if you can get a weight on a small animal, we try to get a weight. There are some ratios you can use. You've seen us on the lift. We'll measure the length of the animal, and then we measure around the animal. That ratio can be a good indicator of whether the animal's healthy or not, whether it's been eating enough. And really that size of the girth, which we'll talk about next, is an estimate of liver size. The liver in a shark is critically important to storing lipids and all sorts of other nutrients, and is really one of the biggest signs of whether a shark is healthy. There have been people who also use blood as an indicator of body condition. And what they do is look at ratios of different fats that are traveling in the blood. So the ratio of triglycerides to cholesterol. But for the most part, this kind of measurement system is what's been working for us. Next slide.
So the liver in the shark also has really important functions as far as keeping the shark buoyant in the water, right? So they're very oily, which is where those lipids that she's discussing are coming in. And it's going to help keep that shark kind of buoyant in the water so that it's not sinking down to the bottom or just floating up on top? Because we know sharks have to swim to breathe, right? So it's going to keep them nice and buoyant in the water. So with that, you know, looking at the lipid stores, so what kinds of things can you look at with that?
Yeah, so what I've provided you with pictures here is just at the top is a picture of a healthy shark liver. And basically, the liver of a shark should, when you open their body cavity, it should be the only thing that you should see. It should be just filling almost the whole coelom. And when we do ultrasound on the sharks, we're able to see that as well. We can actually see how far the liver comes back. So one of the purposes of our ultrasound is actually to look at the liver and see if these animals look healthy. Healthy livers are made up of cells called hepatocytes. Those are just liver cells. And they should be absolutely jam-packed with fats or lipid. So the top two pictures that you see is it's a ray with a very healthy liver. You can see you can't see anything else in that body cavity. And on the right is a histology slide of that same liver. And again, you can see it's very clear. That means that it's full of fat. So that's a healthy liver. On the bottom is a ray of the same species where that liver is absolutely shrunken down to almost nothing. It's not that golden yellow color, so it doesn't have lots of fat in it. And on the right, you can see that it's not clear, so there's just not a lot of fat left in it. So this is a very, very thin animal that was very likely sick at the time of death. So next. So as I said, it's really hard to look at the outside of an animal and determine if it's healthy. And one of the big tools that we use is blood. And, you know, we've talked before about the fact that the major samples we take on the lift are blood and muscle. And there's just so much that you can do with it to give you some information about the health of the animal. You know, blood itself is just a complex mixture of different cell types and then really specific plasma proteins that work together with a lot of just critical roles in just body function. So your blood carries all your blood gases, your oxygen and your carbon dioxide. It carries all the metabolites, so all the stuff that you digest in your body and breakdown products of food that you need to excrete. They travel in the blood. All your nutrients that you're absorbing travel in the blood to the different places they need to get. Hormones travel in the blood to the places they need to get in order to signal normal reproduction. Your immune defenses, how you defend your body against illness, all travel in the blood. And clotting, you know, sort of acute traumatic events. Blood is just probably one of the single most important tissues, which we do consider it a tissue, for indicating the health of an animal. And I included this slide in part, for a very selfish reason, which is just that this is the first white shark blood that I ever saw. So this was white shark Montauk, who I sampled in 2016 off of New York. So this was one of the young of the year.
So can you explain to the kids, they have learned, depending on where they're at, different types of blood cells and how blood might function or impact our body. So how similar would this type of blood be to our blood as far as the cell structure goes?
Sure. So it's similar in that it does all the same jobs. So you have red cells for carrying blood gases. You have white cells to help with immune defenses. They're different in the fact that, as you can see on this slide, see how there's that little blue circle in the center of all those pink cells? Those are red blood cells. And in mammals, they don't still have a nucleus. So that's one big major difference. As far as white blood cells, they have white blood cells, but they don't look the same as mammals. And we're still trying to figure out what the exact functions are of some of those cell types. So it really is an active place of investigation.
Thank you. So on the next slide, again, we're going to go into more tools that a veterinarian use, and this one's blood work. So go ahead.
Sure. This is just all the tests that we do, and I mostly wanted to list it out in parts so that you know the tests that we're doing, and we consider this just a veterinary panel. This is the same type of panel that would happen to your dog or cat if they went to the vet. This is honestly the same type of panel that's going to happen to you if you go to your doctor. It's just we're using the same tools, trying to come up with different information to kind of inform us a little more, and then using that information as a conservation tool. So things like your hematocrit or your packed cell volume and your total solids, those are things that I run on the boat and give me a quick indicator of how healthy an animal is. Complete blood count, so counting how many red cells is in a sample and white cells in a sample. The differential count we'll talk about a little more, what types of white blood cells are present and why that's important. Chemistry panels tell us a little bit about organ health and whether our organs are functioning appropriately. And then protein electrophoresis and acute phase proteins are a little more advanced, but really what they're trying to give us a snapshot of is our immune function. and whether we're being stimulated in some way by a stressor or an infection. So I always get asked this question, how much blood can you take? And the reality is there are limits, and we do calculate them, and we particularly calculate them in small animals. Now, admittedly, when we're dealing with some of those really huge sharks that we deal with, it's probably not a concern because it would be really hard for me in 14 minutes to take the maximum amount of blood. But I think what's important is, sharks are very different from mammals. And, in mammals, your blood volume is twice what a shark has at that same body weight. So that's the first thing that's really important to consider. So we have this calculation where it's like the maximum amount of blood volume you could take is 10% of blood volume. Realistically, I take about 0.1%. I take a very small sample. So we have ways of just easily estimating that. But you have to take other things into consideration. You have to take into consideration whether it's a healthy animal and whether they can handle having you take a sample. You also have to take into account life history. We'll talk about that a little later, but not all sharks are the same, and not all of them have sort of the same numbers of cells in their blood, and that has to be a significant consideration when you're taking samples. And then the next really important thing that you have to consider is that sharks make new blood cells way slower than we do. So all of that goes into calculating what's the right amount of sample to take. And all of that actually happens well before we go on expedition. Everybody submits their requests for samples, and we go through and see how much people ask for, and then we decide whether or not we can take that amount from an animal. And I will tell you that we absolutely, in the past, have gone back to people and said, I need you to come up with a smaller sample that you need us to take. So we're always taking the animal into consideration first. So next slide. So this one I put in there just because we're so different. I talk about how, mammals make blood cells way faster than sharks. So the big difference is really the presence of bone marrow. So our bones are obviously very different than shark bones. We know that sharks have a cartilaginous skeleton versus a bony skeleton, which we have. And one of the main functions of our bones is actually to make our blood cells. So the center of your bones is filled with what's called red and yellow marrow. The yellow marrow is predominantly fat, and the red marrow is where your blood cells are being made. So on the right, you see an actual kind of histologic picture of a bone in a mammal, and those are all the red blood cells that are being made inside that bone. So if you compare that to, if we go to the next slide, there we go, to a shark. And what I wanted to say about sharks is sharks do have a lot of the same organs that we do for making blood cells. So as humans, most of our blood cells are made in the bone marrow. Some of those cells go to an organ that's in our chest right here that's called the thymus. Those are usually lymphocytes that need to be educated so that they can protect us from certain types of infection, and that's where they go. And then the spleen is another organ that's kind of in our belly that's there to kind of cycle out older cells and kind of protect us from older cells that may have taken stuff in or are degenerating. So sharks have those things, but they don't have any bones that can handle bone marrow. So they have these other organs. There's one called an epigonal organ that all sharks have. And I put in a picture for you at the top just so you can see it. It's kind of that long pink thing, and it sits right next to their ovary or their testicle in their belly. And that's one of the primary locations that we get blood cells from. And then the other one is the Leydig organ. Leydig organ is only in certain species of shark, and it's right around the top of the esophagus. It's just a little band. So in some ways, sharks are the same as us, but in other ways, they're very different and have different ways of making blood cells. We can go next.
It's interesting seeing that they have similar organs that are just different, through evolutionary processes, how it all kind of just, it still works out, but they're just so different depending on where they're at. It's really interesting.
Yeah.
So the blood collection sites, I think, is one of the most valuable resources for the lift, just being able to get those blood samples because of where they're located and the sites that they have to take them from. So if you'd like to explain that. And just so you guys know, when she talks about caudal fin, that's their tail, just in case. Okay.
Awesome. So yeah, I think most people are used to seeing me take a sample like this, which is taking a sample from the bottom of the tail. There is an artery and a vein that runs in a special channel right underneath their spinal column. that we are able to access by going through the bottom of the tail. In addition to that, there's some other locations. One of them is what's called the dorsal sinus. So there are 2 dorsal fins. There's a little pool of blood under each one of those. That's another site that we can put a needle into to get a blood sample if we need. And then if you're a flat elasmobranch or a flat shark like a ray, we can actually get blood from anywhere along their wings in addition to the tail. So there's just multiple places that we actually can get that sample. So why am I always going for the tail? Well, we found through some other studies that site actually does matter. So even though a place like under the dorsal fin might be a little more accessible, easier to get to, we found that there's big differences. So like that packed cell volume I talked about is 8% lower than what you would get if you stuck them in the tail. And there's other blood tests that are actually lower if you take it from that sinus. We think it's because it just is sitting there as a pool and it's not flowing through the body actively, and that's why we have those differences. So that's why you'll always see us taking samples in a shark from their tail or in a ray from their wing or their tail. We can go to the next one. And I just wanted to show you some pictures of all those different places. So the top left is sampling a little young of the year white shark just from the tail. They have that little tiny stubby anal fin, and I will say that that's probably the best spot. If you push it aside, there's a little pocket there that you can stick your needle in really easily. Right below that is an example of another veterinarian I work with, Rob Jones. He's taking a sample from a big sand tiger shark from the side of the tail, that caudal fin. This is something that we've been trying in white sharks, in part because I do think there's going to be a day when we get one so big on the lip that we're not going to be able to roll it over, and I'm not going to be able to get under its tail. So we're actually accessing the same site that we do when the animal's upside down. I'm just going at it from the side. So literally there's this little channel where it's normally I'm coming in this way. In this way, I'm coming in from the side. So same site. The top right, I'm just showing where you would go if you wanted to get into that dorsal sinus. So you literally just lift up the little tail on the dorsal fin and go right underneath. And then this is a photo of a friend of mine taking a sample from a ray at the New England Aquarium. And literally they have these little rays that go out from their body. They're called ceratotric. And in between every single one is a little tiny blood vessel that's supposedly easy to access. I've been told you can stick a ray anywhere in the wing and you're going to be able to get a sample. That is not true. That is not true.
Question here, Harley, from a viewer. A lot of these photos are from, they look like they're from animals in an aquarium. How many blood samples are being taken from aquarium animals versus animals in the wild?
Sure. So some of these photos are going to be from, in aquariums in managed care. And I would say a lot of aquariums now have really strong veterinary teams. And the other thing that you add to that is the fact that a lot of these animals, even though people don't think of sharks this way, are trained for certain veterinary behaviors. So we have animals in our aquarium that are trained to voluntarily swim into a stretcher. If they swim into a stretcher, we can easily roll them over and we can easily get blood samples. So all of the animals at the New York Aquarium, the sharks, they all get an annual exam. They all have an annual physical and they get blood samples taken. If anyone's sick, they always get a blood sample taken. As far as field projects taking blood, I think there's a lot now that do take blood samples, just because it's such a critical sample for so many different types of tests. Now, whether or not they're doing actual health studies like what I just described, that might be one major difference.
Okay, thank you. I just wanted to jump on that one real quick.
Yeah, thanks, John. So next slide.
So a lot of the other tools that they use are ones that you guys may be familiar with because they're things that you may have interacted with when you go to the doctor's office or have heard about at least. So Harley is going to go over the ways that they use these different tools to help with the health assessment of the shark.
Yeah, I really just included this in there to let you know that sharks are no different than your dogs or cats or even people. We are able to do all the same tests that you can do. It's just a little challenging because, sharks need to be in the water. And in many cases, they need to be under anesthesia in order to do these tests. So you've seen us do ultrasound. We definitely do that. If you've ever had an endoscope done to maybe look at the inside of your stomach, we can definitely do that. We can take x-rays. This is a horned shark that's in the New York Aquarium. collection that we took an x-ray of because it wasn't feeling well. And, there's even people who've been able to do CAT scans and MRIs on their sharks. So, the sky's the limit. Just because they're sharks doesn't mean you can't do all those things.
Well, I got a question. This isn't from a viewer. This is from me. I know whenever I've had x-rays, they tell me, be still, don't move. I'm assuming you have to make sure the shark is not moving.
And in many cases, in the case of this horn shark, this was a animal that was very easy to anesthetize. So most of our animals are done under anesthesia.
Okay, that makes sense. I was just very curious as to how you might do something like that.
Sure. So now getting into the tools. So, blood counts. There's lots of different ways to, assess blood. Some are very simplistic, where you just look at kind of the ratio of cells to the ratio of the proteins that are present. Other ones are a lot more labor intensive and use a microscope. So you're looking at the exact number of red cells present, the exact number of white cells present, and, platelet counts. Sharks don't have platelets. They have a special cell type called a thrombocyte, but it serves the same purpose. And then also just looking at the types of white blood cells present, because that actually gives us a little snapshot on whether or not an animal is healthy.
One more quick question here, Harley, that came in that I think is a really good one before we go into the tools again is Liam asks, is there a difference in values of the blood between wild versus aquarium sharks?
That is a critical question that we're trying to ask. Obviously, as an aquarium veterinarian, knowing normal in the wild is super important when you have animals that are in managed care. And so we're often just trying to determine that to make sure that those counts are the same. So there's going to be basic things that are the same, the things like their red blood cell counts, their white blood cell counts, they should be the same. I think the big things that are often different are some of those nutritional markers that if you watched what we were talking about yesterday, those can be very different just because diets vary when animals are in managed care to what they're able to catch for themselves in the wild. So I was done with blood counts. Sorry, John, what do you need?
Oh, I just clicked the wrong button. I meant to advance it and I popped back up. Surprise.
I thought you had another question.
Nope. All you. Oh, I went the wrong way.
We'll go forward. Going forward.
So before we get into it, can you give them a little definition? I've heard you use some of the other, terms with the hematocrit. Can you explain to them a little bit what that actually is so that they can follow along with this next one?
Yeah, sure. So, I put up both names for this test, hematocrit or packed cell volume. I think packed cell volume is going to be the easiest one to understand because basically we put a tiny sample of blood in that tiny little tube and we put it in a centrifuge and we spin it really, really fast. And what happens is all the cells, the red cells all settle to the bottom, the white cells settle to the middle, and the plasma stays on top. So that's what we're measuring. It's just like a basic ratio of the different cell types that are in the blood to the amount of plasma that's present. And it gives us like a little bit of information about how the animal's doing. And that's pretty much what this slide demonstrates, which is what I just talked about. So basically the erythrocytes or the red blood cells get packed on the bottom. The buffy coat is actually all the white blood cells and all of the thrombocytes or the platelets in that middle bit. And then you have the plasma on top. So the next slide shows us a little bit of information we can get. So here I gave you kind of like a representation of what we can see where the white blood cell count or the buffy coats are a little bit different. So we know that the one on the left is probably about normal. Like that's about what it should look like. When we look at the one in the middle, we see that that's way more than it should be. So we're suspecting that this animal is going to have a high white blood cell count. So this is one way we can get that first snapshot of health while we're still on the boat. The one on the right is an animal where we aren't seeing a white blood cell band in there. And that might also mean that the animal is sick because it's not making white blood cells. So all of that kind of just very simple information still gives us a little quick picture of the health of the animal. If we go to the next slide, I'm talking about packed cell volumes here just to kind of, give you a sense of the fact that all species are not the same. So not all species have the same number of red blood cells in their blood and circulating at the same time. So mammals tend to be around a little more than 40%. That includes us. And that even varies by gender in humans. Birds tend to be a little bit higher, and reptiles tend to be very low. So you think to yourself, so where do like fish and sharks fall in all of this? So we can go to the next slide. Whoops, go back one. So what we know about sharks is it's not all the same. So sharks have very different lifestyles. And, I don't know that we've talked about lifestyles in these lectures, but, when we talk about white sharks, we're talking about what's called a regional endotherm. This is a shark that can actually maintain a body temperature that's slightly more than the water. And they tend to have the highest packed cell volume. So they tend to have the most red blood cells circulating. They tend to be really powerful, active animals. Other fish species that are like this are things like tuna, animals that have a lot of, fast swimming speeds, high intensity swimming. When we talk about pelagic ectotherms, these are sharks that live out in the ocean, but can't maintain their body temperature any different than the ocean. So they just kind of live at whatever the ocean temperature is. This is things, like blue sharks. And they tend to be sort of in between. They tend to be in the 30s, as far 30% of how much they have in the way of red blood cells in their blood. And then we have what we call benthic ectotherms. These are the sharks that sit on the bottom. These are the nurse sharks that you see, or the bamboo sharks. These are animals that don't have a super busy lifestyle. They're kind of the couch potatoes of the elasmobranch world, and they have the lowest number of red blood cells when you spin them down. So I think what we can get from this whole picture here is when we're looking at these blood samples, lifestyle is going to impact how many red blood cells you have circulating. But if I had a, white shark that had a packed cell volume of 20, that's going to be very abnormal. But if I have a nurse shark that has a packed cell volume of 20, that's actually normal for the animal. This is just kind of my way of saying that, we really have a lot to learn about these animals, and that's why it's really important that some of this, even just basic stuff, that we establish what normal is. So the differential count is where we start getting really kind of down into the nitty-gritty of looking at the blood smear slides. So I do make blood smears on the boat. I will tell you over the years, I've tried very hard to read them on the boat, and I will tell you that I really can't, because it's really hard to use a microscope when it's going back and forth like this. So I tend to save most of my slides until we're at dock for any sort of reason. So basically what we're looking for in a differential count is the types of white blood cells are present. So it's not just the number, whether it's a high or low count, but what types are there. And the reason that that's important is because sharks on the whole are very secretive about whether or not they have an illness. And whereas we, when we get sick, might have high white blood cell count, we're finding more and more that sharks don't necessarily respond as quickly in that way. So when we're looking at health indicators that we're used to using in mammals, that may not be really working out in sharks. And that's why we've started going to this sort of reviewing of the blood smear in order to see what cell types are present. So if we go to the next slide. So in order to determine what cell types are present, you have to actually define what those cells look like. And I think people think that we know what all the shark blood cells should look like, and I will tell you they are all over the map in what they look like. So every time I'm looking at a new species, the first thing I do is put that slide on there and drive around for a while, and I go, okay, what does this cell look like? What does this cell look like? And I'm just trying to define normal. And this is very much what I did when I looked at my first white shark. And these photos are the photos that I took when I was like, okay, this is how I'm going to define each of the blood cell types for white blood cells in white sharks. So that going forward, this is how I always define them. So when we talk about a differential count and the types of cells that are present, really, first we need to know what's normal in sharks. They're a little bit unusual in that the lymphocytes are present at higher amounts than the rest of the cells that have granules in their cytoplasm. So the way that I've divvied these up, that top row there are cells that have little granules in them. And those are what we call granulocytes. Those tend to be the ones that are fast actors. They get there first if you have inflammation and infection. So if they're present at high numbers, that means they're probably responding to something. And that's one of the reasons why we look to see how many of those there are. The ones on the bottom, the monocyte and the lymphocyte, those tend to be a little more slowly acting. So all of this kind of information when we're looking at a blood smear is how we start trying to tease out whether an animal is sick and whether it's having a response.
Harley, I know that you specifically have taken blood from the young of the year all the way up to some of these mature sharks. And people always discuss how our immune system kind of develops over time. Did you notice a difference in the white blood cells in the young of the year versus these mature sharks?
That is one of the questions that I did ask from this whole study, and that's been the real advantage of being part of such a big, complete project where we're looking at all the life stages, is that we're able to ask those kind of questions. And it does look like they're very consistent across that age class, and there aren't big differences in either the number of cells present or the types of cells present, and that's probably good. It probably means that these young of the year are not responding to something yet.
Okay, just wondering.
Yeah, no, that's fine.
So looking at a chemistry panel, this is another really cool study that they do on board, looking at the different, chemistry is what's going on. And we spoke with Lisa Hoops as well, and she has some really great Lisa Crawford has really cool information on some of this as well in prior talks that we've done. So can you go ahead and explain some of the things that you're specifically looking at?
Yeah, absolutely. So when you spin down blood and you get that kind of clear component at the top, which we were looking at, that's the plasma. And that's where all of those metabolites I talked about that are coming from your food, your organs, how you're basically using energy and making wastes. That's where all that stuff lives. It's also where all your electrolytes are. So this is where we get a good picture really of how organs are functioning. So we basically run a panel of 17 different tests. It's very, very similar to what you have done when you go to the doctor that gives them the same amount of information. And what we're looking at is things like, is the liver functioning correctly? There's a couple analytes on there. If you look on the right, there's one called AST and GGT. Those give us a sense of whether the liver is functioning correctly. Kidney is super important for managing things like electrolytes, also excreting waste. So we're looking at a lot of stuff on that other side. We're looking at things like sodium levels, chloride levels. We're looking to see that there's the right balance of those things. It's, lots of different analytes can be used in different ways to kind of assess function. Muscle metabolism, we're looking on the right side at something called CPK. That gives us a sense of how the muscle is functioning or whether there's any damage. And again, like I said, electrolyte balance, it's really important in sharks. So all of this gives us kind of that snapshot of health. But that requires having a normal reference range. And I think, everyone will realize that I can't just, go to the literature or go to a book and open it up and go, oh, what does normal white shark chemistry look like? We're literally writing the book right now by getting the samples that we're getting. And it's also important to recognize the fact that these are relative normals. So obviously, if a shark was out there swimming in the ocean, and then it came over and it gave you a blood sample voluntarily, that might be a little bit different from if you have to, bring it to the boat so that you can sample it. So we're taking all of that into account when we develop these normals.
So as you're talking about that, there was a very good question that came in here from Freya. And she asked, do pregnant white sharks have more / different blood than non-pregnant sharks?
Yeah, so it's not different in kind of cell types. It's more different in some of the proteins that are present, which is literally what we're going to talk about next. So we do know that, and also hormones. And we aren't really talking too much about, reproductive health in this lecture, but a lot of the stuff that we look at in the blood that's going to be different is hormone levels, and then some of the proteins that are present, and I can show you on the next slide. I think proteins are next. Yeah. So there's hundreds of different types of proteins that are present in that clear part where I have the arrow, the plasma. And again, they're important for carrying metabolites. They're important for, maintaining equilibrium with the environment, immune response, clotting. They're also critically important for sharks, like as I told you, in that their white cell counts have not been as helpful as they are in mammals in telling us whether or not they have a disease or whether they're having a response. So we've started looking harder and harder at the proteins that are present to see if we can get a little better idea of whether an animal's healthy, because shifts in the types of proteins that are present actually happen before the white blood cell counts change. So you're even getting kind of an earlier picture of something changing. So we can go to the next slide. And this is an actual example of something that we do. So basically, we try to separate the proteins in the plasma into big groups. And basically, when you run it through an analyzer, it separates into 5 different little bumps. And by seeing what percentage of the proteins are each of those, we establish what we think is kind of the normal amount of each of those, what we call fractions, to have. And that's where in female sharks, that fraction 4 tends to be much, much higher, particularly if they're kind of in an active stage of reproduction, than a male. So that is someplace that we will see a major difference. The other types of proteins that we're looking for, I've listed these out. They're very specific, but they're called acute phase proteins. Again, these are proteins that our liver makes to respond to an infection or a stress long before we ever see changes in the white blood cell count. So this is another thing that we're looking for in the plasma of sharks to give us a better sense of whether they're healthy or not. And the photo on the right is an actual test result from one of the animals that we have in this study, which is white shark yeti. We sampled that animal in 2016 off of Nantucket. I just wanted to show you, that all these tests are being run, and this is what they look like. So we can go to the next slide.
So the next few kind of things we're going to talk about are more things that can impact this health. So looking at nutrition, reproduction, and environmental factors. So these are, ways that their outside area can really influence the health of the shark. So looking at nutrition, just like with us, sharks also have to have a good, proper diet and healthy nutrition. So what are some things that can kind of cause some issues as far as their nutrition goes?
Yeah, the purpose of these next slides is really to show you that the work that I'm doing is not in isolation, and that there's a lot of other studies that are really important to understanding health for an animal. So all these blood studies that I'm doing where I'm trying to understand, are the organs functioning correctly? Are the blood cell counts, correct? Are they showing signs of inflammation? Are all impacted by whether the animal's in good nutrition, whether the animal is reproductive or not, and whether or not they've been exposed to toxins. Super critical. So as far as nutrition goes, I know you've had other lectures that are much more detailed, but the big thing for me as someone who studies conservation is if the nutritional status of that animal changes, why is it changing? Is it changing because they can't catch the prey that they like to eat? That could be due to changes in habitat. That could either be due to human stressors or environmental changes. Have they changed the way that they are foraging, so looking for prey items? And why is that changing? Is it changing because the water is warmer where they used to go? Because humans have come in and changed the environmental structure? So those are big ways in which nutrition can be impacted by climate and humans. So they're also important to consider. Go to the next. Again, reproductive health is something that we do on all these animals. We need to better understand their cycle. We don't know what parts along the Atlantic coast are important to them from a reproductive standpoint. Where are they having their babies? Where are they breeding? We don't know those questions. And the only way we're going to know that is when we handle them by asking questions like, is your ovary mature? Are you pregnant? Those types of questions are really important. Again, we talked about the fact that we do blood work looking at the same exact hormones that we look at in humans to determine, whether they're pregnant, whether they're mature. These are all important questions that we do ask of the blood. And then, you had some great lectures from Lisa, who's a good friend of mine, Lisa Crawford, just talking about the impact of the environment on health. And she talked a lot about exposure to heavy metals and toxins, and the fact that nutrition, we talked just yesterday about the fact that nutrition impacts that. A lot of this is dietary exposure. A lot of it is lifetime exposure through the diet, so it accumulates over time. that third point I have there is really the big question that we're all working together to answer is like, what is the actual impact on health? If the animal has a high mercury level, is it actually making it unhealthy? I think those are things that we're still trying to answer. And we're doing that by taking the health parameters we just talked about and trying to correlate between all of those questions, whether, if I'm having a shift in one test, whether they are nutritionally healthy animals, are they reproductively active animals, are they showing signs of inflammation? Those types of questions we're really still trying to work out. So, this is really just kind of my final summary slide, just talking to you about using health as a tool to do conservation work. And really the problems that we run into is that lack of baseline information, and why it's so important to have groups like OCEARCH that can give you access to big animals that are really hard to handle, so you can get samples that you otherwise couldn't get and get that baseline. We also have a really limited understanding of how the environment and how humans are impacting these animals. And only through doing the studies that we do are we able to start really getting a good assessment on how we really are impacting their lives, both individually and as a population, and affecting their health. So for our group, one of those solutions is we want to understand the health status of sharks. We want to know if the white shark population right now is healthy. We want it so that we have something to compare to in the future. We also want to investigate the effects of different stressors. We want to know, like I said, from an ecotoxicology standpoint, if they are bioaccumulating toxins, is that an actual stressor? Is it making the animal ill? What impact is that having? And this all comes back to identifying shark populations that are at risk. We know that sharks are incredibly threatened by fishing and harvesting. We also know that they're incredibly impacted by habitat destruction. So the question is how is their health also impacting these populations? And as I said at the beginning, their ability to respond. Because it's all well and good if we put policies in place to kind of protect animals from certain fishing practices or stop habitat destruction in certain areas. But if that population doesn't respond, what else is going on in them that is impacting their ability to do that?
So something really cool from, a high school student standpoint is Dr. Newton really is the one getting all of that baseline data. And if you keep following along with what she's doing, you get to watch that grow and, evolve into something that could potentially be, groundbreaking science. And that's something that a lot of you can actually, pursue as a career and take that baseline data that you just heard about right now and potentially, have some discoveries as well. So it's really cool seeing that whole process happen.
Yeah, Harley definitely has one of the coolest jobs, and I think everybody on the ship has respect for what she does, because she's also, as the veterinarian, she's closely monitoring the shark, making sure it's healthy while it's in our care, on top of while she's doing it, because like she said, everything we do is designed around making it as safe for the shark and the crew as possible. And so Harley plays a big role in that, and obviously it turns out she's a pretty great teacher too, so we are super lucky to have her on board with us. If you guys have any questions, please use the comment box right below to ask them now. We can take just a few. I hope Ryan's question here isn't about to make me look silly, because I don't know what this is. What is oncotic pressure? Is that ONCOTIC?
So what is oncotic pressure? Yeah, so that was on one of the slides that I meant to edit that out. Basically what it is sharks try to maintain their blood to be, hyperosmotic, to have higher solutes than the ocean. Obviously, they live in an environment that has lots of sodium and chloride and other electrolytes in it, which is very different from the life that we live when we're in the air. It's also very different from animals that live in fresh water that don't have a lot of those other elements. So one of the major things that's very different about sharks is that their blood has much higher and different levels of different electrolytes and other metabolites that are able to help them maintain that pressure from the environment.
Excellent. Okay, I'm glad that Ryan didn't get me there.
No, it's
Or
just
you.
I meant to simplify. Sometimes as a veterinarian, I don't remember.
All good. Anyway, while we wait to see if any other questions come in, I have a question for you, Harley, if you don't mind. What are you most excited about looking forward as you continue your research?
Oh, I mean, for me right now, the most exciting part is that we have enough animals that we can actually start doing something with the information. Not just for myself, running statistics on this information and trying to separate animals out by age classes and see if there's any differences or where we catch them and whether there's any differences, but also working with other people. Yesterday's live stream is probably the most fun that I've had in a long time, because it was really one of the first times that myself and the two Lisas sat down and really started putting all of our data together and being like, hey, so, how does all this stuff associate? Is there anything that I'm finding that might help inform your research? I mean, we were literally sending whole spreadsheets of information back and forth to each other just to see how stuff associates. And, this type of collaborative research is present in lots of programs, but for me, OCEARCH is my first experience with that, having such a huge team with, there's 22 different projects, and, I don't have to do it all. I can focus on my one little area because there's someone else who's a specialist who can ask questions in their area, and then we can get together and ask questions together. So that's what I'm looking forward to, is that we're finally getting to the right number of animals to be able to do stuff.
Another question that just came in here, and you may have answered it a little bit, but I'll ask it a little bit more directly. Is white shark health changing as a result of the pressure that humans might be putting on them?
The answer is we don't know. And, for me, I am literally just setting up a baseline for one of you in the future to come in and fill in my footsteps. 20 years from now, 30 years from now, 40 years from now. That's how we're going to be able to answer that question, because the reality is we don't have a lot of those baselines. A lot of the things that I thought would be simple to just find in the literature, just find papers on, just don't exist. Simple things. So that's what we're doing. We're focusing on creating normal, and I'm doing that so that one of you in 40 or 50 years can go on an OCEARCH expedition and maybe do the same exact study and ask that question.
Thanks. That question was from Tian. Josie has a good question here. Say we do find out that the sharks are being stressed somewhere. Will there be anything that we'll be able to do to help them?
I mean, that is the primary reason that I think we're all here doing this research. It's great to learn this information, but it has to do more than that. And that's where science-based policy comes into place. All of us are really working to ask these questions, to identify stressors so that appropriate policies can be put in place. So if it's things like we need to regulate what chemicals are allowed to be used or released into the environment, I mean, these are all things that come from this primary research. It's even the simple things, even the movement information is so critical for doing habitat protection. If we can't prove that an area is critical for a life stage of an animal, then we can't protect that area. So all of the work that we're doing is really just to go that next level up and put, smart policies in place. And I say smart policies because I think sometimes people are afraid of regulation and policies because sometimes they feel very restrictive. And I think all of us are really interested in putting policies in place that balance our needs as humans and, commercial fisheries with the needs of the animals. You have to have that balance. They can't be unbalanced.
As a high school teacher, something I suggest that's very similar to this is we read Silent Spring with my environmental science students. So if you wanted to read up a little bit of things that have happened prior in the past, with DDT spraying and the impacts that did and a lot of information that came out of that, I highly recommend reading that if that's something that you're really interested in learning about and something that is a similar situation, it's just something that has already happened versus what we're trying to figure out now. So definitely pick that up for a summer read if you're interested in that kind of information.
A very, very good question, Josie. And then I think we'll make this our last question as we start running out of time here. Just a quick refresher. Melissa asked, you said that shark cells are not similar to our cells. Do they have a nucleus? Was her question.
Sure. So what I was saying is that, when you look at mammals like us, our red blood cells don't have that little blue spot in the center. They don't have a nucleus. They shed it as part of the process of becoming a red blood cell. When we study other species, birds, reptiles, fish, bony fish, sharks, they all have that nucleus. So I was more pointing it out because there are a lot of folks who maybe at this point have had the opportunity to look at blood and would realize right away that that slide looks totally weird compared to what it would look like if it was a mammal.
Excellent. Some very good questions that came in there. We love seeing great questions like that. At least one of you tried testing me, but it was no problem for Harley, obviously. So thank you all for your participation there. Jen, Harley, thank you guys for taking the time to put all of this together. Certainly some really cool work happening, and I'm so excited to see what happens, like you said, as you start comparing it to some of the other your work to some of the other work from other collaborating scientists, because I think some really, really cool stuff is going to start happening here pretty soon. So thank you to everybody who joined. Before we sign off, Jen, Harley, any last things you want to add?
Just wanted to thank you guys all who have stuck with us this whole time. We're kind of coming to the end of some of these lecture series for the little COVID break here. So appreciate you tuning in with us and interacting with us. And always remember, if you want more information, to go to OCEARCH.org, click on the education tab, and you can get a bunch of free resources available for you to do a lot more research and investigations along with us. So thank you again, and have fun this summer if you're able to be done. I know we're done this week, so enjoy it.
Yeah, and I just want to thank everyone for giving us the opportunity to teach and, making this break something really special. I've never had the opportunity to share my research too widely, and I think, trying to share it with kind of the next generation, the folks that I know that are going to come in my footsteps, is really important to me. So I really appreciate you guys sitting through my talk today about veterinary medicine, and hope that it'll maybe plant a seed in some of you about the fact that, you can go to vet school and you can learn all about dogs and cats, and you can actually wind up instead working on sharks.
Yeah. All right. Thanks, guys. I think we're pretty much out of time here, but we will keep all of, we will have more live streams like this in the future, not as regularly necessarily, but stay tuned to all of our social media channels, and we'll let you know when you can get follow-ups to some of this research and learn about other exciting shark-related stuff. So I think with that, we will sign off.
Bye.